Sorry for the delay. Shit, just been working really hard. So, please forgive me. Yeah. But I got here, didn't I? I haven't even eaten anything today, so I'm going to eat some Cheez-Its. I hope that's okay. And, how's everybody doing? Anybody? Anybody here? Barry, what's up, Barry? Still drinking your beer, man. I really appreciate the beer, Barry. You know, it's a nice way to finish the day. A dinner of Cheez-Its and a beer. Keeps me going strong all day long. Man. So how's class going? Finish up all the experiments yet? Is that crunching really annoying? If so, I'm sorry. I'll stop eating Cheez-Its. Yeah, the melon was pretty good. The melon cart? Yeah. I didn't mind it at all. I'm more of like a, lighter beer type of person. I like something refreshing. I'm not a big IPA fan. It's like, why drink something that has to taste bitter? Doesn't make sense to me. I want it to taste good. So anybody in the class who's taking the class, you got access to a lab. What kind of lab? What kind of lab is it? Like an academic lab? I just, the world is my lab, I kid. Oh, man. So, I was hoping people would come in today with their questions about the class. But it's just being you, Barry. What do you want to talk about? Oh, nice. Where do you live at, Barry? Where's the community lab at? Hendra Virus Gaming. You live out in the middle of nowhere? Where do you live at, Hendra Virus? Where's the middle of nowhere? Maybe I'll just, oh, San Diego. Nice. Which community lab are you working with down there? Oh, yeah, the La Jolla Library one. I didn't check out the La Jolla Library. I think I almost did one time, but I haven't been there. Yeah, I imagine so. They had another space down there. I can't remember the name. I'm surprised though San Diego doesn't have more, in terms of biohacking, with all the biotech around there. Hendra Virus Gaming, IL Galva. Is that Illinois? Israel? I guess everyone I know, right? Huh. I'm surprised. Yeah, there's not more people. But yeah, everybody probably works at a company in San Diego and La Jolla, or university, Scripps or Illinois. Where's Galva, Illinois? So I grew up in Illinois. I grew up in the suburbs of Chicago and went to school at Southern Illinois University in Carbondale. So where is this Galva? Is it in like the yeah, where is it? What big cities is it close to? I hate the lag on the YouTube question answer. It's so annoying. Like ask a question and um, you don't get to see a response till like 30 seconds later. YouTube needs to fix that. Like it can't be that hard to reduce that lag, right? It's probably a combination of like the lag of the video combined with the lag of the chat. It's between Peoria and Davenport. All right. All right, hit me, Barry. Hit me with your question. You want to isolate a gene from an unsequenced species. What do you know about the gene? Do you know anything about it? Uh, how do you know you want to isolate it? Silk gene from an antlion. What is an antlion? Antlion. Well, I mean, if you know absolutely nothing about this organism, like absolutely nothing, um, you're just going to have to sequence the whole genome probably. If you know something, like the silk producing gene is conserved or something like that in some way, what you can do is you can use what they call degenerate primers. So a degenerate primer is just like a primer that would match the gene from like a known species, but um, the the gene you leave room for like some mistakes and errors and stuff like that. That's a possible way. You can look for extremely conserved regions in silk genes, um, and then PCR and sequence from those conserved regions. Uh, yeah, if you don't have any conservation based on the sequences of other organisms, you're in a tough spot, and you're probably just going to have to sequence the whole genome. But um, you know, nowadays it's not even that expensive. But yeah, it's a lot cheaper just to sequence genes. So you probably order some primers first and um, see if you get any hits, get any luck. And it's similar enough to other silk genes. Justin said the other classes that are offered. So this 101 class is a really good class. It's for people who don't have any experience with biology in general, hands-on. In all of our classes, we ship you all the materials you need to do all the experiments you need to learn for the class. So this class teaches you everything from like no experience to be able to genetically engineer back microorganisms and use CRISPR on microorganisms. It just, it gives you like the groundwork to do more complicated stuff. Then we have a 102 class, Bioengineering 102, which teaches you how to like edit and program DNA. So that class is a little more advanced and a little more, well, it's a lot, it's pretty advanced. We've been having a little trouble with it because, it's complicated. I don't know if we've had anybody actually complete the final experiment yet. So there's that. Maybe we should make a prize for somebody who completes the final experiment in the 102 class. We're running a Genetics 101 class, which teaches PCR and sequencing and analyzing DNA sequencing and submitting DNA sequencing to databases and all that good stuff. That class it requires a PCR machine, so it's a little bit more expensive. So in 102, the final experiment is to purify some DNA, 2 different pieces of DNA, and then swap the genes between the pieces of DNA. So they're fluorescent genes. So one is a red fluorescent gene and the other is a green fluorescent gene. So you're swapping the two. Yeah, it's just it's a little more technical and tricky hands-on, but and then we got our human cell culturing and engineering class, which is really fun. We just started that, but you can still sign up if you want. Where we teach you how to grow human cells, and from human cells you can grow organs, tissues. Knowing how to grow human cells, you can also know how to grow animal cells. So if you want to try to make a push or some effort into clean meat, or growing chicken cells in a petri dish, growing beef cells in a petri dish is totally possible. Yeah. And we are starting a mushroom growing class where we teach you how to grow any mushrooms. You can even just go buy a mushroom from the store and grow it in your home. So that's really cool. Yeah, that class is going to be pretty exciting. Yeah, because like shiitake mushrooms and shit like that are really expensive. And when I started talking to William Padilla Brown, he was teaching me this stuff, and I was like, wow, that's awesome. Um, Barry, when does the next iteration of classes start? I don't know because we're gonna have to wait till after the holidays because the holidays are just so busy for everybody. But we're gonna have the classes, recorded versions of the classes already up, so you can take the classes as you want as a recorded version. Um, so that will be available. But yeah, I don't know. Um, we'll see. Maybe February, maybe later. Um, yeah. It's just too hard to run classes November, December, and even January because of all the holidays. And yeah, the mushroom one is just pre-recorded, so it's not going to be live. Um, it'll just be a pre-recorded class. But yeah, we're trying to increase the offerings of our classes. It just takes time to develop the class. Um, so, but yeah, I think if it were me, I'd probably take the 101 class and then the human cell class. Um, the 102 class and the genetics class are more specific. Um, that it's a specific skill that if you need to learn it, you need to, and if you don't need to learn it, you might not use it. So shrug whether you should take it or not. But yeah, the 101 and the human cell class, those are really important. Are there any projects I'm working on that I need help with? You know, uh, I'm a bit a loner when it comes to my scientific projects, uh, because I view it kind of like an art. And like I know there's artists who have other people who like build their stuff, but like that's not me. Like I like to do all the hands-on stuff. I don't just like to have the idea and then somebody else execute it. I like to be part of, you know, the execution or be the execution. Um, and so that's a big part for me. Um, if you're interested in projects, I don't know, Barry, are you on Facebook? Are you in the Facebook group, the Biohacking and Genetic Design Facebook group? If you're not, you should join that Facebook group. Um, there are a lot of people with a lot of projects, and I'm sure a lot of people need help. And so, going on there and interacting with people there, I'm sure you could find somebody who you can collaborate with or work with, who could teach you a thing or two and also, use your help. So, no Facebook. Just make a fake account, man. Make a burner account. Come on, you know? Do I have Slack? Yes, I have Slack. I just don't use Slack a lot because, it's just not part of my everyday life. If you want to focus on making some transgenic plants, so Justin, what I would do on our website, we sell a lot of the stuff. But the first step that I would do, if you've not really done any cell culture, um, is learn how to uh, do plant culture, cell culture, plant culture, all those things. If you haven't already. Um, on our website, we sell a lot of the materials and chemicals for plant stuff that you'll need. Um, I don't know if we still have a plant section. You might need to just search it out. Oh yeah, we do. We still have a plant section at the top. Go to the plants. Uh, we have most of the chemicals and stuff that you'll need. to participate in start working towards doing plant genetic engineering. I mean, the first thing, I mean, there's common protocols. I don't know if we have one up. We might. The first thing I would do is learn to make calluses. Um, so I know Sebastian Cocchioba might have some. Sebastian Cocchioba. He's a biohacker who works a lot with plants. And I think on his Patreon, he might have some resources and guides. Let me see. I have to. Ay, ay, ay. Gotta verify my account. Really? Come on. Now it's making me confirm my country of residence. What is going on here? God damn it. All right, let's see. Sebastian. Let's see what he has. Let me see if his Patreon. I thought he had some somewhere. It might. Yeah, I mean, generally, if you're searching for plant cell culture stuff, you're going to be able to find stuff online. The hardest part with plant genetic engineering is, yeah, Sebastian is Bionomica Labs. With plant genetic engineering is the actual genetic engineering. You're either going to need to use agrobacterium, or you're probably going to want to use agrobacterium to make a stable line. And agrobacterium is actually, you need a permit to ship it across state lines in the US. Shipping it internationally, I think, is probably next to impossible because it's a plant pest, right? It infects plants. It's a virus for plants. Or it's a bacteria that infects plants, not a virus. Yeah, so getting a hold of agrobacterium is the biggest thing. I know there's like an underground network of biohackers that you can probably get some of it from. Yeah. So, yeah, there's no link. I think YouTube censors the links you post. Let me see if I could change it. Um, hmm. Yeah, I don't know. I think YouTube censors links. So, just tell us what to search on the internet, or where you found it, or what's going on. But yeah, buy some, go on our website, buy some, do we sell Mershigi Skoog Media, MS Media? No, we don't sell MS Media? Oh, that's crazy. We need to add MS Media to our website. We have MS Media. Yeah, make up some MS Media with, 2,4-D. Figure out how to grow plant calluses. And, you're like halfway there if you can do that. Halfway to engineering plants. Ants. All right, yeah. Put some Murashige Skoog media on the website, but I need to update it. But yeah, that's what you need. Plant cell culture protocols. Yeah, I mean, there's a lot of info out there on this stuff. It's there's a lot of guides and protocols and stuff like that. So um, just check it out. Get started. Do I have reputable biohacker contacts in Canada? Looking for hands-on learning in the Vancouver area. I don't think I know anybody in Vancouver. Yeah, I mean, if you're looking for a lab to work in, the thing is, if you want to get hands-on experience, like I say, buy some supplies from our website. It's a great way to get started, to do stuff yourself. We have some really cheap, inexpensive kits. I think our cheapest kit is like $29.99 or something like that. And then we have like a beginner setup for like $299.99 plus class. That's a pretty good deal. You know, teach you how to do genetic engineering and molecular biology and microbiology stuffs. I think that's the thing with this stuff nowadays is you don't necessarily need somebody to teach you. You can learn yourself, You can That's the great thing about these classes that we run. Yeah. Uh, so what's going on? What else? Any other questions? On all the software. What software? Are you specifically talking about, Barry? Or are you just talking about in general, like software used for molecular biology and the websites used and stuff like that? Um, Snapgene, the NCBI, yeah, I get you. I feel you. Um, we do have some classes on that stuff. I think I don't know how much that's utilized in the 101 class. I can't remember. I know there's we do a little tutorial on Snapgene, I think, in the 101 class. 102, better tutorials, yeah. I mean, yeah, the 102 has a little bit more info. If you watch the first 102 class, so if you go to the Odin YouTube page and watch the first 102 class, I think the first 102 class goes pretty in depth with Snapgene, talks a bit more about it. and spends a lot of time on it. But yeah, a lot of these things are just intuition and understanding what to look for and the way things need to go and work and stuff like that. And that's it just takes time. It takes time. Yeah, there's no easy way to skip that. I think in like class 7 or 6 for 101, class 6, there's a nice instructional about using a bunch of different software and websites. But yeah, not really tutorials, but at least not so much. Exactly. It's exactly like learning Photoshop or something. It's just, it takes time. And it's hard to skip through all that time investment. The experience just develops that intuition to help you understand what's going on and what you should know and what you should be looking for and stuff like that. It's tough. Learning anything is tough. It takes time. It takes an investment. So I just, I try to put in a little bit of investment as much as I can. Sorry about yawning. A friend from graduate school was in town last night, so I was up a bit later than normal. And I'm old, so when I'm running on like 4 or 5 hours of sleep, I yawn. Forgive me. It's pretty warm here in Oakland today. Woof. All right, what you got? Anybody got any more questions? Hit me with them. Hit me with some questions before I take off. Because if there's no more questions, I'm just going to go. And I will say sayonara. Was that racist? I'm sorry, I didn't mean to appropriate anybody's culture. Yeah, no problem. We'll still be around. So if you're still working on that last experiment or you still need advice or something like that, ask away. Feel free to, message me on Facebook or whatever you need if you still have questions that you need answered. Adios. Yeah, still be around to try to help answer questions. I'll do my best. You just got to be really annoying sometimes because I'm really busy. Just be like, answer my questions. Yeah. Alrighty then. No more questions. Thank you all for taking the class. Hope you learned a lot. If you have any questions about anything, feel free to post them in the Google Classroom or just ask me directly, through email or I have no idea. That looks Korean in some way. But I have no idea what that says, Justin. I hope it's good. But yeah, feel free to contact me on Facebook or email or anything. Barry, thanks for the beer. You'll always be dear to my heart for sending that beer over to our lab. Everybody here appreciates it. But I couldn't tell it was misspelled, so don't worry. But yeah, thanks all for taking the class, and hopefully you take another, and we meet again. But until then, have a good one. Exactly.